Let's get started. Good morning. Thank you for joining us this morning. I'm Laura Rudberg, MassBio's Director of Events. We're here for MassBio's virtual Possible Talks. These are 15 minutes of inspiring and engaging content in the style of a TED Talk. With me today is Jim Boushell, VP of Biospecimen Solutions at Precision for Medicine, a biomarker-driven life sciences contract research organization working with diagnostic and therapeutic researchers globally. Jim will discuss some of the largest changes he is seeing that affect all researchers and compare these to the shifts over the past century of pandemics to help us understand what comes next. Some examples of these changes include shifts in data needed to conduct research, new approaches to conducting research remotely, new paths to fast-tracking R&D, and innovations in drug repurposing. Whether a researcher, a healthcare worker, or a citizen in affected areas, these changes will have an effect on you. We have reserved a few minutes at the end for Q&A, so go ahead and add those as you think of them. If you see a question in the Q&A that you've already thought of, you can upvote that, and that'll bring it up to the top of our list. With that, I'm going to go ahead and turn it over to Jim. Jim, go ahead.
All right, thank you. And good morning, everybody. And Laura, thanks for making me sound so special. I really didn't think I was going to sound special till probably Sunday on Father's Day would be probably the next opportunity. So thanks for that introduction. So good morning, everybody. Thanks for taking time out of your day to sit in on this webinar. I think we have a pretty interesting topic to discuss or share with you, and then hopefully at the end we'll be able to talk about some answers. So let me get my screen going here for you. There we go. So I think you can see that. Everybody see the map? Yeah, good. Good. So again, my name is Jim Boushell. I'm a Vice President for Precision for Medicine. For those of you that don't know, Precision Medicine is a life science contract research organization, as Laura said, and we specialize in doing biomarker-informed clinical studies. We operate, looking at the map, we operate in more than 25 countries with 35 offices within those countries with all different levels of service, from actual laboratories to offices that sit with clinical teams. Now, my team, we're located right over here in Norton, Norton, Massachusetts. So we're not very far from where everybody is on this call. My team's role in the value chain for Precision for Medicine, well, that's in targeting and collecting patient specimens and data sets. And the reason we're doing this is so that biomarker researchers can do the discovery work and move their products into clinical trials at a very rapid pace. I don't think it's rocket science here or a surprise to anybody, but without specimens, you can't do your research. So whether it's a tissue, whether it's a whole blood sample or a cell, or even if it's just some data, you need it in order to be able to do your research. So why am I here today versus any of you? Well, I've been part of the Mass Biotech Council for almost 20 years now. I've been operating behind the scenes, working with biotech and pharma companies, helping move their programs along. And everything changed for me back in January, as it did for all of you, when coronavirus knocked on our door. Actually, coronavirus didn't knock on our door, it knocked down our door. During that time, I quickly recognized that everything that I'd been doing over the last 25 years with my team here, which was filling laboratories and biobanks with specimens and large data sets, well, that could prove to be a very powerful tool, a weapon in our battle here with COVID. Now, we certainly didn't build any of this with a pandemic in mind, but we did build it. And the fact that we're able to leverage it every day amazes us each and every day. Surely, each and every one of you probably have similar stories with how your companies or you personally have reacted to it. And maybe later on in the talk, we'll have an opportunity to hear something directly from each of you. So the topic of our conversation came about in a conversation that I had with Laura and the team, and we thought, wouldn't it be interesting to look back at some of these prior pandemics and try to see if there's anything from those pandemics that we learned about that we're actually leveraging today in our battle with coronavirus. And you know what? Here we are. We're going to talk about it today. So quick service announcement. If anybody here just realized that they jumped off on the elevator at the wrong floor, it's too late to leave. We own you. Please stick around. We'll be less than 25 minutes here, and I'm sure we'll try to keep this a little bit fun. Switch my slide here. All right, so as I said, let's take a look back at some of these recent pandemics. And we'll just take it 100 years. What we'll do is we'll go back, we'll look at the pandemics, we'll do a little bit of history lesson, touch on some of the key topics related to that particular pandemic, and see if there's any connection between what was happening then to what is happening now. So let's start back in 1918. 1918 was a very, very busy year in history. Locally here, and I don't know how many of you are natives, but we do know that the Red Sox won the World Series in 1918. And we won't talk about what happened for the next almost 100 years afterwards, but it was pretty important to us locally. But seriously, really what was important in 1918 is World War I had come to an end. And at about that same time, the Spanish flu, which many are considering to be the worst pandemic in our modern history, it began its attack on the world. It was a terrible, terrible pandemic. I believe it took more than 50 million lives from our world. The Spanish flu was a really fast-acting flu, so fast that the scientists and the pharmaceutical companies couldn't catch up to it. In fact, the only way they were able to control that flu was using what's called social science back then. Today, we call it social distancing, and it worked, and it did contain it. And in less than 2 years, the virus had left the world. So now let's jump forward. We'll go back to 1957. 1957 was a time where the second major pandemic in the 20th century showed up on our doorstep, and that was in the form of the Asian flu. Now, the Asian flu is considered to be, a novel virus. Novel in the fact that it's part avian flu and part human. Yet, its impact on the world was far less than that of the Spanish flu. Now, what makes the Asian flu so notable, especially in US history, was the fact that we had developed a vaccine for it before it made its way here to US soil. Pretty amazing accomplishment, especially if you think about the fact that it was 1918. And we can thank a young virologist that was down at the Walter Reed Medical Center and his interest in reading the newspaper. In fact, he was reading the New York Times when he saw an article about a virus that was ongoing in Hong Kong. And from reviewing, he was really realizing that it was ravaging children, and the symptoms were very, very alarming to him. He sensed that this was going to become the next great pandemic. He didn't waste any time. He immediately went back to the office the next morning, picked a team of researchers, and he sent them to Japan. And getting into Japan, they went there and they found soldiers that had actually been infected with the virus, drew blood samples from them, and came back here to the United States to do their research. In the middle of their research, they figured out real quickly that the patient's blood samples that didn't have any antibodies. So mounting a defense to this virus was probably not going to happen. And so he immediately took copies of that virus and distributed them to a number of pharmaceutical companies at that time and asked them, pleaded with them to go ahead and start working on a vaccine. To nobody's surprise that knows this person, it took just 4 months. And at the end of 4 months, the vaccine was made. And at about that time is when the Spanish flu made its way to the United States. Now this vaccine, it's being credited for saving over 1 million lives, and it's probably even many more than that. Okay, so let's jump over to 1980, or the 80s in general. It was a great decade. Big hair, big bands, and big problems. Big problems coming in the form of HIV. And yes, HIV is a pandemic. HIV is a retrovirus. It's not a flu. It's transmitted via direct contact or through transfusion. It's not through a respiratory particle or anything of that nature. A cough and a sneeze is not going to give you HIV. HIV was different. It was different, and so was our response. The way that we approached this scientifically was we weren't going to measure it based on whether or not we eradicated HIV. We're going to measure success based on whether or not we can manage or control the progression of HIV and its symptoms so that patients didn't go all the way over to what's called AIDS, or what's known as AIDS. Now, how did they do that? They did it by testing on different treatments and identified a product called an antiviral, specifically AZT. And what they realized is when they administered AZT to these HIV patients, the virus in their bodies actually settled down. However, unfortunately, what they found was it quickly responded and came back up and started taking over the body again, potentially leading to AIDS. So over the last 30 years, lots of research has been done and clinical trials have been done on testing different levels of antivirals and different combinations of antivirals in the hope of being able to control the virus. Well, as we all know today, it works. So this combination therapy, it's known as a cocktail, and it really is the earliest form of personalized medicine. And because it's personalized medicine, it essentially requires constant monitoring, much like some of the therapies that we're doing now in cancer and other areas. The way that they did it with the HIV patients was they gave them the cocktail, the patient went home, they lived for a while, they come to the doctor periodically, had their blood drawn, and when they had their blood drawn, the doctor sent it out for what was called a viral load test using a technology called PCR. I'm not going to get too much more technical with it, but essentially what happened was if the virus stayed the same or it decreased, everything was good. There was no reason to do anything or change anything. However, if the viral load had increased, well, that changed the game, and they realized at that point in time that the patient's body has changed itself. Actually, the HIV has changed itself in the patient's body, and they need to manipulate that combination of therapy, that cocktail, and make it so that it doesn't. And so when they do that, they just change it. They add a little bit more of this antiviral, a little less of this antiviral, or they switch them completely out. But essentially what we learned from this is the fact that we can live with a pandemic amongst us, and specifically here with HIV. So here, during my research, I came across a lot here in looking at viruses and pandemics, and I learned quite a bit. Came across this quote from Hippocrates, and I never thought I would actually share a quote from Hippocrates, but I will. And it states that you declare the past, you diagnose the future, and diagnose the present, and then you foretell the future. In a nutshell, basically it says that history is teaching us a lot. And these pandemics, well, they didn't really disappoint. And I skipped over some key points that we're going to touch right now and tell you how we've learned. So remember the Spanish flu? Yeah, remember how fast it worked and the fact that scientists couldn't catch up to it? Those were scientists in 1918. And around 2000, 2003, here in the United States, there were a group of scientists that were really curious. They wanted to see what was going on with this virus. Why was it so impactful? How come it killed so many people and spread so quickly and disappeared so quickly? So what they did is they set out to reconstruct that virus. And they did that using a technique called reverse genetics. And through reverse genetics, they were able to rebuild that virus and then test it and analyze it with many different technologies and try to figure out what's going on. And what they determined was the root cause for this issue was a protein, and a protein called polymerase. So with that information in their bag, they turned around and they started testing in the labs with antivirals. And what they realized real quickly was that those antivirals were stopping the replication or slowing down the replication of the virus, thereby making the condition less significant. Now let's draw that all the way up to current times. No surprise, when coronavirus hit our doors, everybody said, we have no treatments, we can't do anything, what are we gonna do? We leaned on an antiretroviral, okay? And that was remdesivir from Gilead. Oh, wait a minute, went the wrong way. Okay, sorry about that. So let's jump over to the Asian flu. So the Asian flu, if you recall, I said was a young virologist. The gentleman's name was Maurice Hillman. They described him as being both brazen, they described him as being brash, and albeit probably bold too. Rumor has it that his 5-year-old daughter walked into his bedroom one morning with a sore throat and swollen glands. And like any good dad, he jumped up, he cultured her throat, he took it, put it in a bucket, and brought it to the office. Well, guess what he developed? From that swab and that virus, he developed the mumps vaccine. So, guy's a little different, but you know what? We can thank him for it. So what did he do here with the Asian flu, and why is it important to us? Well, as I mentioned before, when he found out that there was no antibodies to it, he contacted those pharmaceutical companies directly, and he had them working on that. He basically bypassed the US vaccine agency in an effort to get the treatment to the patient, to the population sooner. Thereby, it's a historical version of fast tracking the vaccine. You know, today, we're seeing it in real time, right? The government's partnering with industry now. So they're doing it in the form of emergency use authorizations, it's EUAs. They're streamlining that pathway so that we can get treatments and diagnostics into the hands of those that can do work with them and not create more bottlenecks. So that last pandemic, the current one, HIV, we're still living with it, gang. But what we have figured out is that through a combination of behavior and biological modifications, that we can coexist with it. Okay, this next slide is just a fun one. Not a lot of content here to get to you, but if you look at it, there's two images here. The image on the left, well, those are auto workers in Detroit in 1918 during the Spanish flu. You can see them all garbed up with their masks on. The image on the right, well, those are auto workers also, but it's 2020, and they're making the masks that are going to go out to the population. So that's one of those squirrel moments. I apologize, but I found that real interesting. Hope you did too. So hey, enough about history. Let's talk about some innovation. Let's talk about things that we are doing today. And yes, I know we're early on here in the global crisis. However, we're transitioning the way that we're researching and fighting the virus. We're coming at it with an army of scientists from every different specialty. We've got biologists, epidemiologists, we've got chemists, we got social scientists, and we got the really smart guys, those computer scientists, to help us make sense of what the heck is going on. We also tool them with new weapons. Whether or not you know it, the internet is a weapon, and this is the first pandemic that we're going at it with the internet by our side. We're utilizing social networks. So there are people monitoring them just to see where some of the shifts of changes, infections, things of that nature are going on. In the laboratory, we're taking on these unique technologies, or these very highly sensitive technologies, such as next-gen sequencing, to help us understand what's going on with the virus and to monitor whether or not it's changing. And then finally, artificial intelligence. With all of this work going on, lots of data, a lot of information, a lot of points coming in, there's no way a human being could do it in a quick enough time to actually respond with either a treatment or a vaccine. We saw what happened in 1918. So really, gang, this is all real and it's exciting. And I have to admit, I feel like I'm one of the luckiest guys in the world to be able to have a front row seat here of it. So as I mentioned, COVID is a global crisis, and we're studying it that way. We're studying it globally. There's people all over the planet here working on it. And 100 years ago, we used all that data simply to just figure out how many people might die during the pandemic. Today, we're using that data in real time. We're breaking it down and we're trying to identify what are the predictors of survival. We're trying to figure out who are the people that are most at risk. And then we're also accelerating and developing new vaccines and therapies just from that information. At Precision, we're making sure this happens for our clients. We've leveraged our clinical trial infrastructure. We've taken our labs and our investigators globally, and we've focused them in on collecting and finding patients with COVID-19 for our clients. We've also taken our technology platform known as Quartz Bio, and we've repurposed it so that we can harmonize all of these multiple data points coming from every walk of life and every part of the earth here and digest it and give us the necessary insight that we need in order to identify biomarkers and potential drug targets. So like all pandemics, COVID-19's given its all. It's really trying to prevent us from beating it. It sent our scientists home, away from their labs, traditional resources and teams. It's keeping patients from getting to the doctors. So how do you do a study if you can't get to the patient? Frankly, it's done a pretty good job at trying to stop us. But like you, we didn't give up. We improvised. For us, we followed the governor's ask. We sent our essential staff home, our non-essential staff home. We kept our essential staff here in our facility so that we now picked up additional space and we're able to keep our laboratories open and work in a safe environment. For our studies, we were able to keep those enrolling also. Utilizing that staff that we sent home, we developed a new strategy, a strategy that wasn't going to be limited by the fact that we couldn't get to the patients through the doctor's office. What we were doing is we were sending out information and messages using technologies such as social networking and the internet so that patients could find us. And then we had to develop the teams to go out and go meet them at their homes and conduct the study visits. Bottom line is, it's enabled us to continuously support our scientific partners ever since day one. And more importantly, as these programs evolve, we're able to adapt as we learn more about this virus. Now, if time allows, I'd like to spend some time at the end here and hear some of the things that you've done differently. All right, so biopharma. Biopharma has long been looking for ways to accelerate drug development. Coronavirus was just that urgency that we needed. As a life science community, we didn't disappoint. We mobilized, and in fact, we did it overnight. As I said earlier, government did their part. They accelerated access. They provided us the EUAs. But all of the industry, all the academia, they did their part too. They were submitting thousands upon they were sharing data by submitting thousands upon thousands of papers to preprint servers, and in fact, overwhelming them. All of this providing the needed insight, to identify potential on-market therapies that we could retool and repurpose and use in this fight. With Precision, I know this is real because we're actually doing it too. There's a local example here in a company called Karyopharm. We're running a clinical trial right now for them in Europe of their anti-cancer drug, selinexor, on COVID patients with severe acute respiratory syndrome. So each of these novel methods we talked about, they're transforming how we're able to do our research today and combining it all real time with surveillance methods to know where the virus is going and where it's been, as well as whether or not it's mutating. And at the same time, when we bundle all this information together, our techniques, our science, what we're really figuring out is that we're allowing scientists to really shine. We're allowing them to uncover therapies for the ill and develop vaccines for those at risk. And along the way, we're making COVID-19 history. Okay, so I've spoken a lot here and I'm going to sum this up real quickly. I don't have a crystal ball. I can't tell you what's in our future, but what I can share with you is that science and medicine, they're incredibly powerful forces. And the way that we're bringing them together today, it's making us better armed to not only defeat COVID, but any future pandemic. So with that, that's the end of my presentation and I'll turn it back to Laura.
Thanks, Jim. I have to say I'm a sucker for a documentary That is my go-to on Netflix. I mean, I enjoy all the fluffy stuff too, but I am a sucker for a documentary, so I love having this in historical perspective. I really appreciate that you gave that back story. We do
Sure.
have some questions already. The first is, Jim, can you share how your company is able to recruit COVID-19 patients and, if applicable, collect and annotate blood or nasal samples? Safety concerns and privacy issues would seem to be challenging.
Oh, that's a lot of questions.
Yeah,
So
it's a lot.
I will break that down. So first question was, how were we able to conduct sample collection?
Yep.
Okay, that's
How do
number
you,
one.
well, the first question is, how do you find the patients? How do you get your hands
Okay,
on people that you know had
absolutely.
COVID? Yeah.
Absolutely. So, how do we know that people have COVID? Well, what we've done, as I mentioned earlier in the presentation, we've deployed a different strategy, one using the internet and using social media. And so, what we do is we make ourselves visible, lack of a better term, and what we're looking for and what we need. And so, we are putting out not truly an advertisement, but more of a knowledgeable piece of information saying, if you have COVID-19, you may be interested in participating in. So that's one way we were able to do it. So patients can reach back into us and contact us and participate. Another way to do it, we form partnerships with some of the area neighborhood health centers with the walkthrough clinics and things of that nature. Again, everything we do is done as if it's a clinical, well, everything we're doing is a clinical trial. So we're operating under what's known as an IRB approved protocol, which basically dictates how we interact with people. And we're obtaining informed consent at that time. So when the patient comes through the clinic or comes through the testing area, we can then meet with them later with their caregiver and tell them a little bit about our study and potentially enroll them. What was the
And
other
then
half of the question?
the last piece
Oops,
was safety and privacy concerns. How do you manage that or mitigate those?
Sure, absolutely. So I'll touch the first one right off the bat. So privacy concerns. Again, we operate in accordance with what's known as good clinical practice. So this is a clinical study. We handle everything in that manner. So if someone's consenting or any of the personal identifiable information, it never makes its way back to us. It stays with the caregiver. Okay. If it's someone that we've identified through our social media campaigns and they actually come into us, they come in online to us and they ask to be part of our studies, we'll then do a consenting process that lets them know that we're doing, and then we'll send someone directly to their home. And when they get to their home, they'll do the actual study procedure at that point in time. Now, when you're sending someone to a home or you're meeting with the clinic and we're dealing with coronavirus, the PPE or the gear and the equipment that you have to wear is far different than what we were wearing 6 months ago. We've equipped our teams. Safety number one. We won't do the collection if we can't ensure the conditions. But here we are to date. It's been a successful program, and it's continuing on.
Awesome. Question 2. And you had said that one of the things that you folks did was you sent home non-essential employees, and that made more space for lab employees. So I can see how that probably helps. But what have you found to be the biggest challenge in keeping your business functioning during the pandemic? I'm sure there's a million, but what's the biggest one?
Yeah, I'm gonna, and this probably isn't the answer the person that question asker wants to hear, but it's morale. It really is. It's we're asking people to come into work every day when they hear on the news at night or when they're driving to work, whatever, and the governor's saying, stay home, everybody locked down. And that is obviously important advice, and I'm not saying don't follow it, and we certainly didn't tell our team not to follow it. What we committed to our team was that we would make sure they're as safe as they possibly can be. We wouldn't put them in any scenarios where there's unnecessary risk, and more importantly, that we're constantly connecting with them. So I'm not a laboratorian, I'm not sitting in the lab, but I did make it a point and have to be in the office every single day since this thing hit our world in the middle of January, along with my lab team. And that's for morale. That's to let them know that we're on board here with them. We're making sure that everything's controlled, and we're making sure that they are also in a good position. If there's an employee that really isn't, maybe it's weighing heavy on them or they're really nervous, by all means, we tell them they can go home and we can figure this out afterwards. But, for us, it wasn't necessarily that we were open and everything had to happen at the normal rate that it was prior to it. For us, it was more like we need to stay open because there's a lot of research going on, and those researchers need the patients and the specimens, which is what we provide in the data. And without it, we basically it would create a bottleneck for R&D. And so it's just being conscious of who your team is and who your people are and being open with them.
Makes sense. All right, one last question so far, unless anybody has more questions, go ahead and throw them in. We do have 3 minutes, so we can probably get another 2 questions in. What types of data are essential to attach to COVID-19 biospecimens? So what are the essential data points? Data points?
Yeah, sure, data points. So obviously, right now, COVID-19 is being studied. There's two different levels of studies. One level of studies is you're just trying to figure out how you diagnose it sooner. So if you're doing a study and you're trying to diagnose it sooner, then the samples and the information that you capture has to be related to the patient's health at that point in time. So I think there's 16 different data points we capture, all related to symptoms. Symptoms from headaches to nausea to I'm not going to tell you whatever that may be. You want to get their body temperature. You want to know what that is. You want to know where they've been, how they've been doing it. You want to know how recently was did they have how far back was the onset of those symptoms. So that's that acute patient. What we call the recovered or the convalescent patient. That patient, the information is nice to have, although it's not absolute to have. So when we're bringing them in, we're capturing everybody with the exact same data set. So whether they're acute or they're convalescent, we're capturing all that data because what we figured out over time, and again, we were involved in the HIV pandemic early in the mid-90s. What we figured on time is information is key. So we try to get as much information as possible.
Yeah, I'm sorry. There's like questions flying in now. Everybody heard they had 2 minutes left, so now there's more. I have one in the chat here. In the chat, come back up, chat. Where did you go? There we go. From Milan. How has your company implemented remote monitoring to run clinical trials during the pandemic?
Oh, it's a great question. You're going to ask me that. I have a full clinical team that makes me look really smart and good, and I didn't have them as part of this program. But I can tell you what I do know because I do sit in on the meetings. Investigator sites, clinics, whatever. We're not going to have that same level of accessibility. So remote monitoring is necessary. We've developed a whole new set of procedures to be able to do that, and began scheduling with each one of our investigator sites, set blocks of time where they're going to have staff on their end working directly with us in a controlled environment where we can share information electronically and conduct as much of the monitoring visit as we can. My feelings are there will be a point in time where we're going to have to get on site with most of our sites, if not all of our sites. But I would be speaking far outside of my wheelhouse if I gave any set dates on that.
Last question, because we have just a minute.
Okay.
Any advice to other researchers whose programs are still shut down?
Call precision. No.
I
promised I wasn't going to do that. No, if your program is shut down, do as much of the research as you can, do as much work as you can from your home, and then lean on some of the outsourced partners that are available that are still conducting essentially studies to feed your research, and then figure out how you're going to do it. Restarting your studies are probably going to be the biggest challenge once you get back inbound. And that's just going to take some thoughtful planning, but there's enough companies like ours out there that have more or less worked throughout it and developed procedures and processes that are working, that another month or so we'll be sharing all that information, and hopefully we can make it easier for everybody to get back on board and get back in the labs.
Awesome. Well, we appreciate all the work that your team is doing to help us get to what we hope is an end result of some vaccines and some treatments, which hope that's forthcoming soon. Last weekend, right before we closed last week, I said Happy Father's Day to everybody. That was not last weekend. I cannot keep track of time during pandemic.
This weekend.
So Happy Father's Day this weekend to all the dads out there. Don't forget to call your dad if he's still with us. And everybody have a super weekend. Thank you, Jim.
Terrific.
Really appreciate
Thank you.
your time.
Thanks for having us. Everybody have a good weekend.
Bye, everybody.